All right, welcome back. This is the second half of Chapter 11. We are continuing our discussion of cancer treatments, and of course, we can't leave out chemotherapy. So, chemotherapy consists of drugs, chemicals that either kill cancer cells or block their division. So, most of these are going to act by either blocking DNA replication, if a cell can't copy its DNA, can't continue through the cell cycle to a division, or they will interfere with the mitosis process themselves. And so, in this sense, these drugs are going to be toxic, are going to kill any dividing cell, right? Any cell that's actively progressing through the cell cycle. They're not going to be specific to cancer cells. And that's what causes some of the well-known serious side effects associated with chemotherapy drugs. Hair loss, nausea. So in the case of hair loss, you have dividing cells in your hair follicles. In the case of nausea, you have dividing cells in your gastrointestinal tract. You've got dividing cells in your bone marrow. So, in worst case scenario, these drugs are causing nausea, diarrhea, anemia, immune deficiency, hair loss. So those are not trivial side effects at all. So as we'll talk about, some of the newer cancer treatments are really designed to try to get around some of these side effects and make cancer treatment a bit more specific. But I do want to talk about the five main classes of sort of classic chemotherapy drugs. These are the anti-metabolites, alkylating platinating agents, antibiotics, that may be surprising to you, some plant-derived drugs, and then hormone therapies. And I'm just going to briefly go through each of these groups just to give you some idea of how they work. And we'll start with anti-metabolites. And you see in the table here, the majority of these are pyrimidine or purine analogs, which would basically act to block DNA replication. And so a lot of these would be considered anti-metabolites. So these are going to be substances that mimic, normal molecules involved with cellular metabolism, and they're basically blocking the action of those molecules and hence poisoning the cell. So one example here of an anti-metabolite is methotrexate, this guy right here. And this will bind to and inhibit this enzyme dihydrofolate reductase, which would normally convert dihydrofolate into tetrahydrofolate. And this is required for synthesis of some of the nitrogenous bases that you need for DNA replication, right? So a basic metabolic activity that's important for progressing through the cell cycle. So if you treat with this anti-metabolite, you block that process. Right? The cell is not making those nitrogenous bases that it needs, and DNA synthesis is effectively blocked. Or, as I mentioned, a lot of the chemotherapy drugs in this class are nitrogenous base analogs. So you can see down here, the normal bases on the left, here and here, and then the analogs on the right. And so these analogs have a similar enough structure that they will basically fool DNA polymerase into grabbing them and incorporating them into a growing DNA strand. But once incorporated, now they're going to block either addition of further nucleotides, or they're going to disrupt the normal structure of the double helix enough that they will hopefully trigger apoptosis. So those would be the anti-metabolites. Then we have the alkylating and platinating agents, and these are basically all DNA cross-linkers, meaning they will insert themselves in between the two strands of DNA and then covalently bond to either strand, hence cross-linking those two. And if the two strands of DNA are cross-linked, that's going to prevent them from being easily separated and prevent, again, DNA replication from occurring normally. You can see all the alkylating agents. They have these two arms. These would be things that were first developed really as chemical weapons, something like nitrogen mustard gas. But it was later discovered that they could also be used to kill dividing cells intentionally. Platinating agents, work in a similar way. They're so named because they contain platinum at their core. But yeah, what I want you to know, these are cross-linking agents, block DNA replication, and hence progression through the cell cycle. Then we have antibiotics, and of course, we usually think of antibiotics as something we're going to take to kill bacteria rather than cancer cells. But there are a number of antibiotics that have been shown to have some chemotherapeutic utility. And in particular, these are antibiotics that would work by either cross-linking DNA in a manner we were just talking about. They might intentionally induce double-stranded breaks, or they could inhibit this enzyme called topoisomerase. There's a few examples of antibiotics working in that manner. And if you've forgotten, topoisomerase is an enzyme that's important for DNA replication. It basically works ahead of the replication fork, and it will prevent excessive pressure from building up as you separate the two DNA strands. That pressure could result in breaks, double-stranded DNA breaks, which could be pretty catastrophic for the cell. So the topoisomerase will relieve some of that pressure and prevent those breaks from happening. So if we inhibit that enzyme, we're encouraging those breaks, which would then hopefully lead to apoptosis and death of the cell. Then the next group are the plant-derived drugs, which are just so named because they are originally isolated from plants. There's some very famous old chemotherapy agents in this class, things like Taxol and vincristine. So these are, quote-unquote natural drugs, although, to be honest, the vast majority of drugs that we use were at least originally derived from plants. Sometimes we then figure out ways to synthesize them in the lab. But the majority of these sort of traditional plant-derived chemotherapy drugs either act by inhibiting topoisomerase, as we were just talking about with some of those antibiotic chemotherapy agents, or they will act to disrupt microtubules. So they can bind to and inhibit microtubule polymerization. And that's significant because microtubules make up the mitotic spindle, right, which of course is what's moving the chromosomes around during mitosis. So if you disrupt the spindle, you disrupt the mitotic division. And then lastly, we have hormone therapy, which is arguably probably the most desirable out of these 5 classes because hormone therapy is going to sidestep a lot of those side effects that I was talking about a few minutes ago. Is going to tend to be a lot more specific, is not going to destroy normal cells typically. But using hormone therapy really only works in hormone-driven cancers, which tend to be in specific tissues, breast tissue, prostate tissue, some lymphocytes. Their division tends to be very hormone-driven. So you can think of this as being useful for cancers whose growth is really fed by excess hormone signaling in this case. So just to give you a few examples, there are certain breast cancers that are due to overexpression of the estrogen receptor, right? So they're receiving too much estradiol, estrogen signal, and that's causing those cells to divide too much. So a chemotherapy agent called tamoxifen binds specifically to that estrogen receptor. You can see it shares some similarities in terms of its structure to estradiol, but it does not activate that receptor, so it basically just binds and blocks it and prevents that hyperactive signaling that was occurring in those tissues. Another strategy would be to, instead of blocking the receptor ligand type interaction, is to actually step backwards in the pathway and try to block the synthesis of the hormone, the signal itself. So there's things called aromatase inhibitors that could block estrogen synthesis, right? And that could again, reduce that estrogen-driven cell division. And there's, of course, very similar situation on the male side of things with androgens like testosterone, which can stimulate, in particular, prostate cancers in men. Um, Early treatments were basically physical castration to try to remove the source of androgens. Obviously, that's not ideal. So, luckily, they've come up with some quote unquote like chemical castration techniques. So you could use something like leuprolide, which will block the production of these peptide hormones, these gonadotropins. The gonadotropins, in turn, would stimulate testosterone androgen synthesis. So basically, by treating with leuprolide, you're blocking the production of testosterone. Instead of having to physically remove that source, you're doing it chemically in this case. And just like we were discussing with tamoxifen and estrogen receptor, there are drugs that could block the androgen receptors on prostate cells and interfere with that signaling in a similar way. And there's even similar techniques that can be used with some of the lymphocytes that tend to be hormone driven, resulting in some of the lymphocytic leukemias. So glucocorticoids famously can reduce cell division and reduce the growth of some of those cancers specifically. So these hormone therapies are very attractive for these specific types of cancers, but they're not going to be a cure-all in terms of treating some of the non-hormone driven cancers. So as you may be aware, unfortunately, it's pretty common that cancer cells will develop resistance to chemotherapy as treatment progresses. And there's a number of ways that cancer cells can do this. Um, One very famous is multi-drug resistant cancer cells. And what happens in these cells is they are basically upregulating and activating these pumps, essentially. So plasma membrane pumps or transport proteins that will literally just pump any chemotherapy agent that's taken up by the cell out of the cell. So obviously, if the drug's not getting in, it is not going to be effective. Um, you know, Another issue is that, as we saw when we were going through some of the different types of chemotherapy drugs, some of these drugs basically act by essentially damaging DNA or cross-linking DNA. The idea is that that kind of damage is going to induce apoptosis to cause the cell to be killed. But as we've discussed, right, a lot of cancer cells will mutate or disable the apoptosis pathway. So if you have an altered apoptotic response and you're treating with a chemotherapy agent that's designed to damage DNA and induce apoptosis, that's no longer going to work. Some cancer cells will reduce their ability to take up the drugs. So just we were discussing them pumping chemotherapy agents out, they may reduce the ability for them to get in the first place. Or if you think of something like, let's say, tamoxifen, right, which was going to bind to the estrogen receptor on the surface of the cells and act in that way, what if the cancer cell mutates that receptor so that the tamoxifen no longer binds, but maybe estrogen still does, right? Or maybe the receptor is mutated so that now it's always active, even in the absence of an estrogen signal. In that case, tamoxifen isn't going to do any good. So again, unfortunately, there's a number of different strategies that cancer cells use to avoid the effects of some of these chemotherapeutic agents that we've talked about. And another interesting idea that's kind of gained some traction in recent years is the idea that tumors may actually contain what researchers have called cancer stem cells. So this would be a very small population of these cancerous cells that have, I guess fair to say, like the most potential for division. And so the idea would be, unless you actually manage to kill that small handful of cancer stem cells with your chemotherapeutic treatment, that tumor is very likely to regrow. If you leave even, basically even one of those stem cells in the body. So, how can we deal with this? Well, one approach that researchers discovered pretty early on is treating with several different chemotherapy drugs in combination can be a lot more effective than just treating with a single drug. Especially if those drugs have different mechanisms of action, then it becomes very difficult for the cancer cell to evolve ways around these multiple simultaneous treatments. Um, Another limiting factor traditionally in terms of chemotherapy was the fact that if you treated with these agents that were causing death of dividing cells at a high enough dose, you would actually kill the dividing bone marrow cells, right? The ones that are producing new blood cells and immune cells, and you would leave the patient basically totally immunodepleted. And this approach is still used in some cases because it was realized, well, we could, if necessary, actually go ahead and do that, right? Treat with that incredibly high dose of chemotherapy, uh, hopefully kill all the cancer cells. Yeah, we also kill the bone marrow and leave the patient without an immune system, but now we can go in and basically do a bone marrow transplant to replace those lost bone marrow cells and reintroduce the immune system back into the patient. So that's pretty drastic, but it has been used in some cases successfully. A little bit more subtle, more modern approach is to try to look at gene expression profiles in different cancers to try to gain an understanding of, well, how are these cancers different from each other, and which gene expression signatures are going to respond best to different chemotherapy, different drug combination treatments, so that you could try to tailor some of these chemotherapy treatments to have the best effect based on the person's specific cancer type. Now, I wanted to shift here at the end and talk about a few different emerging treatments. So these would be, newer treatments outside the traditional surgery, radiation, chemotherapy. And one that's really become arguably the most exciting in recent years is immunotherapy, which is trying to use the body's own immune system to attack cancer cells. And some primitive version of this was realized even back in the 1800s. Physicians noticed that some cancers would seem to regress and sometimes even go into remission when patients developed a secondary bacterial infection. And what they realized was happening is, okay, the heightened immune response trying to deal with the bacterial infection is allowing the immune system to also attack the cancer cells, drive it into a remission. So some of the first attempts at immunotherapy, and this is still used in some cases today, was basically taking advantage of bacteria that could trigger an immune response but not cause any serious disease. So something like BCG here that could basically be intentionally placed into a tissue, following a surgery to remove cancer, to try to heighten that immune response again and allow the immune system to attack some of those cancer cells. Another, a little bit more modern take on that approach would be to treat with cytokines. And cytokines are these very potent signaling molecules that are part of the initial immune response. And so they do trigger a very, very strong immune response, but they can be toxic to humans. And you might be familiar with this idea of a cytokine storm. So you've heard that a lot in association with COVID-19, which is basically where your body is producing too much cytokine signaling, and that's basically causing your immune system to overreact and start attacking your body's own tissues. Um, so cytokines can be useful in terms of boosting an immune response, but they have to be used very sparingly to try to avoid that type of overreaction. Um, Another approach would be to generate antibodies against cancerous antigens, right? And try to use those antibodies to specifically target cancer cells for destruction. So you can actually generate what are called monoclonal antibodies. These are basically quote unquote pure antibodies that would just recognize one specific antigen. Um, so there are companies that will produce monoclonal antibodies for you against a specific antigen that you send them. And the idea is if you can identify something that's different, ideally probably on the surface of cancer cells compared to normal cells, now you might be able to generate an antibody that would just recognize the cancer cells. And I don't know how familiar you are with the immune response, but the binding of antibodies to cells in the body, right, can attract other immune cells, which can result in the destruction of those cells. Um, so if you can intentionally synthesize monoclonal antibodies directed against these cancer cell antigens, that could be a very useful and specific approach. Another variation on this theme would be to actually engineer the antibody so that it has some attached toxin or radioactive group such that when it binds to the cancer cell, we don't even need the other immune cells to come in. The antibody itself, because of that toxin or radioactivity, is going to kill the cancer cell directly. So here's an example of using an antibody to treat non-Hodgkin's lymphoma. And so this is a disease of overproliferation of B cells, B lymphocytes. And the antibody is targeted against this cell surface antigen called CD20, which in normal cells is found on mature B lymphocytes, so the little blue rectangles here, but it's not found on the stem cells, the precursor cells that give rise to those lymphocytes. But in somebody with non-Hodgkin's B cell lymphoma, all the cancer cells have that particular antigen. So the idea is you treat with the antibody is hopefully going to bind to and target for destruction all the cancer cells. It will also wipe out all those mature B lymphocytes, but because the precursor cells don't contain that antigen, they're left alone, and they can then give rise to a second wave of hopefully healthy B lymphocytes. And that's just one example, right? So antibodies have been used in quite a number of different cases. As we were talking about earlier, they've been used in breast cancers to bind to estrogen receptors and block that type of signaling. So this whole idea of being able to trigger immunity has kind of brought up the idea of a cancer vaccine. Would it be possible to basically prime the immune system to identify a cancer, or ideally multiple cancers, ahead of time? And there's been some experiments in mice that suggest this could actually work. So primarily this is taking advantage of killer T cells, cytotoxic T lymphocytes, which are a type of immune cell that can bind to cells that are showing some foreign antigen and kill those cells. And so basically in mice, what they were doing is they found they could find some cancer-specific antigens, right? So some protein, something that's unique to those cancer cells, inject them into mice. The mice mount an immune response, develop antibodies against that particular antigen, and then if that specific cancer that contains that antigen intentionally introduced into the mice, the immune system does a much better job at taking care of those cancer cells. So, there might be some possibility for at least some specific cancer vaccines down the road. But really, the big challenge is because cancer cells are our own cells, it becomes difficult to try to find those cancer-specific antigens, and in some cases, you know, they just may not be present, or their difference from the normal proteins may be so subtle that it becomes challenging to take this kind of approach. Um, Another sort of more modern take on this is what's called ACT therapy, adoptive cell transfer therapy. Um, in this procedure, what clinicians will do is they will remove uh, lymphocytes, T cells, from around the site of an existing tumor. The idea being that's probably where you're going to find immune cells that would be most primed to try to attack that tumor. And then in the lab, they would try to screen those T cells, those lymphocytes, for their ability to recognize the cancer cells, propagate those that seem to be best at doing that, and then reintroduce them into the patient. So you're basically, going from a few very active immune cells to a big army of active immune cells. Um, but, there's a bit of a risk here. Well, how do you make sure those lymphocytes are only targeting cancer cells? Because if those lymphocytes are, potentially targeting other cells, other normal tissues, you could have a catastrophic effect when you reintroduce this amplified number of those immune cells. Um, Another even more modern version on this is to remove T cells and basically use some of the genetic engineering technology, something like CRISPR, to enhance those T cells, basically program them essentially to specifically recognize something about a particular cancer, um, and then grow up a bunch of those target cells, reinfuse them into the patient, and effectively let them loose on the cancer. So that's been pretty exciting and has been used in a number of cases pretty successfully. Another approach is to try to use drugs that will prevent cancer cells from reducing the immune response. So, a number of cancer cells have been shown to basically be able to bind to things like T cells and essentially shut them off, deactivate them. So if we could, treat with some drugs that just might simply block the cancer's ability to inactivate the immune system, then the immune system will continue to be able to fight off that particular cancer. Another very popular and more modern approach has been what's called molecular targeting, which is trying to intentionally design molecules, drugs that could target very specific, enzymes or molecules that are contributing to the cancerous state. So there, some of the early ones that have been developed. There's Gleevec, which would bind to and inhibit the specific tyrosine kinase that you find in some of the BRIC-ABL oncofusion driven leukemias that we talked about. IRESA is a designer molecule that'll bind to that epidermal growth factor receptor. So if you have a cancer that's basically being driven by excessive EGF activity, this particular molecule would be very effective. And here you can see just the results of one study. So they're looking at cell viability with various IRESA concentrations, and you can see it's preferentially killing cells that have this mutated EGF receptor, while it leaves the normal EGF receptor containing cells alone. So again, very specific and very targeted. So these are great because of their specificity, but that's also really one of their downsides, because this kind of molecular targeting is only going to work if this very specific molecular target happens to be present in that cancer. So it means, you would need lots of these different molecular targeting agents. You really need to build a library of these to effectively be able to deal with all the different types of cancer that are out there in the world. Um, as these drugs started to be developed, another issue that was realized is in many cases it was just difficult during clinical trials to try to assess how effective they actually were, because of course initially when you have some of these new experimental drugs, they tend to be used just as a last resort in very late stage cancer patients that are already receiving other treatments, chemotherapy, radiation. So in those cases, it becomes challenging to try to separate, okay, what is the effect of this particular treatment versus the other treatments that a patient may be receiving at the same time. And lastly, I wanted to mention that viruses are beginning to be engineered to target cancer cells specifically. So these are called oncolytic viruses. And there's a lot of subtlety and variation in terms of how this can be done, but in general, there's really been two main approaches. So one is you engineer the virus so that the virus will actually enter all cells, right, healthy cells and cancer cells, but it's only going to be able to complete its replication cycle inside the cancer cell. And as it does that, produces new virus particles that would hopefully lead to the death of the cancer cell, whereas the normal cell would not be affected. Or the viruses are engineered such that they can only bind to and enter cancer cells. So they're just not able to attach to normal cells. So they're completely left alone in that particular instance. But yeah, this approach has really been used in particular for specific cancers that have not responded well to some of the more traditional radiation chemotherapy type treatments. Now, if we think of really any of these sort of drug-based cancer treatments, I just wanted to point out all the steps that go into approval of a new drug and the amount of time this typically takes. So, you think about, discovery, research and discovery of whatever the particular drug may be, testing on cells and culture, animal testing, that could be a very long road, 5 to 20 years in many cases. Then if you find something good there, you go to clinical trials in humans, but there's, phase 1, phase 2, phase 3. A lot of times that can take up to 5 to 10 years. If those are successful, then it would go to FDA review, which could take over a year. So, really you're looking at, at the long end, 30 years, at the short end, maybe 11 or 12 years. And that's been in the news a lot recently, because with some of the COVID vaccines being so impressively fast-tracked, right? Because the normal vaccines would be on this kind of time scale in terms of their development and rollout to the public. So the fact that things have been developed in under a year is really paradigm shattering, I think it's safe to say. So, all right, that is it for chapter 11. Chapter 12, which is the last in the book, we'll talk about some strategies for preventing cancers.